Alright, so the next group we want to look at then is the SAR clade. SAR stands for Streminopola, Alveolata, and Rhizaria. So for these three groups then, the Streminopola, we'll be looking at the diatoms and brown algae, for the Alveolata, the ciliates, and for the Rhizaria, the radiolarians. So let's start with the diatoms. So for the study of diatoms, I have a very cool slide here. So here's our slide. This slide was specially made by Josh Stepanek, a PhD student who specialized in diatoms. He went to University Pond, which is right here on campus, went in, grabbed a sample then, and cleaned up the sample and made it so that we could visualize these diatoms really well. So these slides you won't be able to find anywhere else, and really these slides are higher quality than any slides I can get from any other company. Now for the examination of your diatom slide, I want you to turn in your lab manual to page 17. On page 17 you can see photographs. These are photographs of various genera that you'll find on the slide. I'm basically going to work through the slide, not talk much about the slide, but let you look at it. And then you can use these diagrams to identify several different genera of the diatoms. Key thing to note is just the diversity of the diatoms that are present. There's a lot more present than what we have right here on this diagram. So here's a view of the slide under the 4x objective. And I can see a lot of diatoms. So we can see down here most of it is muck, but we can then make out a lot of diatoms. I'm going to increase the magnification to the 10x. I will focus now under the 10x, and your job is to kind of look at these diatoms once we get them in focus and see if any of these match up with the diagrams that we have. So I center that one. Here's a good one. And we'll just focus in. Actually, there's a few in here. There we go. You can see that one really well. So this is under 400x magnification. Okay, it's nice. Easy to identify. A couple more in there that you can actually see. Go back to the 10x. Do another little quick search. And we can focus in. Increase magnification to the 40x. And focus. Some cool ones there. You see the nice little siliceous shells on the outside. Here's a very nice one that you can identify. There, that's a nice image of that guy. Shoot back to the 10x magnification. And I will look for more guys. One dead center there. Put it up to the 40x magnification. And you can see its focus is right there. Nice. There you go. Go back to the 10x magnification. We'll find another one. A little bit different this time. Let's see. What do we have? Here's that guy we just looked at. This one's pretty good. But we already got one of those guys, huh? Okay, another one in there. See that long guy right there to the left of him too, so there's more than one in here that you can identify. Back to the 10x magnification. And you can see that there are tons and tons of diatoms in these samples. So the diversity is amazing. This is just one little pond being Boulder Creek. So this is a very, very, very successful group and dominates Boulder Creek. Now, if these guys were alive, they'd be moving. They move with gliding movement. And so they'd be moving along a little bit, right? Okay, well, that should do it. I think that's enough. So the next group of organisms we want to look at are the brown algae. So brown algae get their name from the pigment fucoxanthin, which is a brown pigment. The brown algae are almost exclusively marine, and they are all multicellular. So when we think back to the original 1969 proposal of the category or kingdom Protista, that kingdom did not include brown algae because that kingdom only included unicellular or colonial organisms. Brown algae then are multicellular. This one is laminaria. It has a holdfast down here that holds to the rocks, a stipe here, and a long blade. Not only is this multicellular, but it does exhibit a life cycle that we'll be looking at in more detail later on in the semester. You don't have to memorize it yet, but it's called alternation of generations that alternates between a sporophyte, this is the sporophyte stage, that makes spores, and then the spores hatch out into a multicelled gametophyte stage that make gametes that are sperm and eggs. Okay, it's kind of interesting to note that the multicellular alternation of generations has been derived in many different times over the course of evolutionary history. We have the green algae, the red algae, and plants all exhibit the same or a very similar pattern of alternation of generations. Now the chloroplast is interesting inside these, or the plastid is interesting inside these guys. Now the plastid in both the diatoms and the brown algae is an interesting plastid because the plastid has a membrane around it with a vestigial nucleus, a second membrane around it with a vestigial nucleus, and a third membrane around it with a vestigial nucleus. And so the thought is that those were made by primary and secondary endosymbiosis events where there was a cyanobacterium. It was then eaten by a heterotrophic single-celled organism that then ended up creating a chloroplast-like plastid inside. It was then engulfed again. It was engulfed again. Okay, so several endosymbiotic events giving rise to the plastid in both the brown algae and in the diatoms. Okay. So the next group we want to examine are the alveolates. And the alveolates then are one of the SAR clade, the A in the SAR clade. And they're comprised of the dinoflagellates, the apicomplexans, and the ciliates. And they're characterized by a membrane-bound cavity below the cell surface called alveoli. Now, what you're going to be doing in lab is that you'll be making a wet mount of ciliates. From that wet mount, you will use this diagram to identify several different genera and put those in your lab report. Then we're also going to look at another one called a Paramecium bursaria and compare a Paramecium bursaria versus a Paramecium multimacronucleatum. Now the next mount I want to make is of what are called ciliates. Ciliates are different than amoeba in that they move by cilia rather than pseudopodia, and we'll examine that under the microscope. So here's my culture. This is of mixed ciliates. Again, it's from Ward's Biological Supply Company. It has a lot of ciliates inside. I'm going to turn it around this time so you can actually see all the debris in the bottom. These guys again are heterotrophic, so they're hanging out in the bottom. And what I want to do is I want to get in and carefully suck up the muck off the bottom. I'm actually going to drop a few drops out so I really get the muck when I put it on this slide. A little bit more here. Okay, there we go. Again, I'll put the lid on. Don't totally screw it on. And we'll get a cover slip. You have to be careful that you don't get more than one cover slip. A mistake a lot of students make is by getting more than one cover slip. There we go, and our wet mount is ready to be viewed with the microscope. Alright, here is our view of the ciliate culture under the 4x objective. And we can see a lot of ciliates. So all of these guys zooming around then are different types of ciliates. You can see the long, dark, worm-like ones here. These two long, worm-like guys there. Alright, so I'm going to search around a little bit and then find some things to try to focus in on so you can actually see the cilia. Still, there are certain types of ciliates that don't move very much, and they attach themselves to basically pieces of debris. There's one of these right in the center. It's called a vorticella. Vorticella are pretty cool because they have a stalk. They'll attach to various devices, but they're still going to act like a normal ciliate. Now, as I mentioned last time with the amoeba is a predator. As a predator, then the amoeba will capture live organisms and eat those live organisms. And we can see that it has a stalk and it's just sitting there. And we can get really close to it here. There it is. And right around its mouth, you can see it has a mouth. Around its mouth it has cilia. Those cilia then basically create a current, and then it uses that current to suck in all of the debris around the outside. 40x on this guy so that we can see him in more detail. Okay, we can see that guy. He has cilia all around. Cilia then are teeny tiny hairs. The key thing about cilia is they beat synchronously. And by beating synchronously, then these guys can have the cilia all around their mouth region. So kind of in the left-hand side, this guy has a nice little mouth region that we can go in and you can see. These guys, I want to focus in on this guy, the greenish guy with the super long stalk there. Okay. It's a single-celled organisms. All of these are single-celled organisms. It means they're one cell that make them up. There are other things in here that we'll look at in a minute that are multi-celled organisms that occupy a very similar niche to these guys. But he's a ciliate. You can see that ciliate around its mouth. Okay, we'll just focus in. There's a vorticella next to it that we looked at earlier. And it basically is similar to a vorticella, only a lot bigger and eats a lot bigger stuff. And we can see it here very well. You can see all that cilia around its mouth. Then the stentor then will beat its cilia. And so here's our paramecium bursaria right in the center under 10x magnification. I'm going to increase up to 40x objective, I guess 400x magnification. We'll center this guy in here and focus. And you'll notice the color on this guy. So first of all, it is green. By being green, then it would appear to be similar to a photosynthetic organism, but it's a paramecium. And the other paramecium we looked at was heterotrophic. This one then has all those little green things inside that the other one didn't have. And those green things are actually cyanobacteria that live inside the paramecium bursaria in a symbiotic relationship. So the paramecium bursaria gives the cyanobacteria home. The cyanobacteria then provide energy for the paramecium bursaria. So the paramecium bursaria then can be both heterotrophic and autotrophic, utilizing energy from the symbiotic relationship with the cyanobacteria. So if you think about all the plastids we've been looking at over the course of this class, how we looked at endosymbiosis occurring over and over again, this would be an early stage in the process of developing an endosymbiotic event. Okay, so here's a paramecium multimacronucleatum, and it is feeding. Let's see if I can't get it into focus here. There we go. These guys are filter feeders, so we can see here, we can see all that cilia around the outside. Basically, he has a mouth. He rams his head into all of that debris, then starts to shoot all of those cilia around and create a current that sucks it into his mouth. Okay, so we can see him here feeding, see him push their heads in, they start to bang around. And then they'll create that current, and when they create that current, then all of that goes into their mouth. So these are paramecium multimacronucleatum. Okay, so the final group that we want to look at in the SAR clade are the rhizaria. For the rhizaria, we'll be looking at the radiolarians. And for the radiolarians, we'll first start with a slide of their tests or shells, and then we will go to a living actinospherium of pseudopodia. Okay, so now we're looking at a slide of radiolarian tests or shells. It's called Radiolarian Ooze is the title on the slide. I'm under 4x or 40x magnification right now. We'll shoot up to the 10x, total of 100 times magnification. And these are really kind of neat. You can see these shells, see all those little teeny tiny itsy bitsy pinholes in these shells in here. Okay. Those are basically what allow the pseudopodia to stick out. Then the pin-like pseudopodia, they can stick all out of all those shells in there. It's actually pretty cool looking. We'll go here, might as well go up under 40x on this guy right in the middle. He looks pretty cool. And you can see all of those little holes where the pseudopodia can stick out on those. Yeah, that's really cool. Okay. Right there. Okay, I'm under the 4x objective, so 40x magnification, and this directly in the center looks like an actinospherium. An actinospherium is a radiolarian-like organism. It's classified in the rhizaria. They're moving up here to the 10x or 100x magnification. We can see next to it is a blepharisma, which is a ciliate, and we can see that we have this central body with all of the little teeny tiny legs going everywhere. Now those are pseudopodia-like legs called axopodia, and those then are used for feeding and locomotion. Now we can see here that we have a blepharisma, which is a ciliate. These actinospherium then feed by basically having sticky components to their axopods, And those sticky components then collect various organisms, and here the blepharisma is either stuck to it, but more likely the blepharisma is actually in there feeding off of the little axopods on the actinospherium. Here you can see that the blepharisma is just kind of moving around slowly, collecting all of those little organisms off of those axopods that the actinospherium is trying to eat. Now there are photos of actinospherium eating a lot larger things, and so there are some photos of actinospherium eating a blepharisma of this size, But you really see a lot more of the blepharisma kind of feeding around the actinopods than you do seeing the blepharisma being engulfed by that actinosferum. So the actinosferum is a predator, but it only preys upon smaller organisms rather than the blepharisma.